पिछले वीडियो में न्यूक्लियस तक हो गया था। न्यूक्लियस तक पहुंचे थे और फैकल्टेटिव हेट्रोक्रोमेटिन तक हुआ था। इस वीडियो में फैकल्टेटिव हेट्रोक्रोमेटिन के आगे से है। फैकल्टेटिव हेट्रोक्रोमेटिन में हम लोग पढ़े थे कि ये टेम्परली इनएक्टिव होता है और ये कभी डाई नहीं किया जा सकता है, लेकिन इसका एक एक्सेप्शन है और वो है बार बॉडी। ये इनएक्टिव, दिस इज इनएक्टिव एक्स क्रोमोसोम इन ह्यूमन फीमेल, which can be observable by dyeing, and it was discovered by Barr and Bertman, so-called Barr body. When staining is done in neutrophil, the inactive chromosome, that is Barr body, appears like elongated rod called a drumstick. According to old concept called as dosage compensation or Lyon's hypothesis, Barr body is an extra X chromosome. It forms from bar body to compensate double dosage of X chromosome. Bar body is used for sex determination and used to identify genetic disorders. Now, one X chromosome is very always mandatory in a human being. It is fixed, and X and other Y would be extra. So, if bar body is 0 and Y chromosome is 1, it is normal human male, and if bar body is 1, that is 1X, and precisely 1X also, and 0 Y chromosome, then normal female. If one bar body and one Y chromosome, then XXY, that is Klinefelter's syndrome. It has 47 chromosome in it. And if um, 0 bar body and 0 X chromosome, then it will have only X and nothing else, because X is mandatory and nothing is present. So this is called Turner syndrome, and it has only 45 chromosomes, only one, and 44 rest. So in female, now in female, if 2 bar body is presented, 0 Y chromosome is present, then it will be X and X. It will be super female. It will have 47 chromosomes. Now, proteins associated with nucleus: histone and non-histone. Histone is basic due to presence of lysine and arginine. These are basic amino acids and are acidic in nature. Non-histone because they are amino acids tyrosine and tryptophan. They both are aromatic amino acids, and they have H1, H2, H2B, H3, H4 proteins. Now DNA coils around the histones protein to form nucleosome. This is the our histone, and this is the DNA. It coils around the nucleus histone to form the nucleosome. Now nucleosome appear as if protein beads. These protein beads are present on the DNA. DNA would be long chain and wrapping on this, so it would appear like this. And if so not, then it forms chromatin threads. It is just like this, appears like this. Non-histone protein role is to that they are enzymatic protein, they are structural protein, and they are regulatory proteins. If there is enzymatic protein, then example is enzyme DNA polymerase. Structural protein example is nuclear lamina. Now regulatory protein, it acts as a signal protein to control the expression of genes. Chromosomes literal meaning is colored body. This is the last topic of this chapter. Structure or body is formed by highly condensed DNA at time of cell division. We mainly study the metaphysics chromosome. We don't study all the in the chromosome. Chromosomes, a big why we study, because chromosome is seen under metaphysics or motifis, shows maximum condensation, therefore best shape to study. Now stage and DNA contents and chromosome number. If the stage is G1, then that is undividing parent cell, that is the normal chromosome, then the DNA content would be 1. C is just regarding the content, and similarly N is here according to number, not a unit or anything. Chromosome would be 1, but during replication the DNA would become twice, of course. Will form two carbon copies of DNA molecule, but then again no change in chromosome. Metaphasic also same, but just the time when these two would be dividing, and they would be forming two sister chromatids. Now fourth would be the anaphase, that is will have separation of this, that would result in one DNA content and uh, one number of chromosomes. Since these two will separate to form two daughter cells, structure of metaphasic chromosome they will have two chromatids attached together or primary construction or centromere. This is the function of them two. Each chromatid has two arms either side of the centromere, this side and this side. Conditions on the length of arm are 2. If equal, then isobrachial, that is this. If unequal, then heterobrachial. Here in heterobrachial, if small arm is present, then it is called P arm, and long arm is called Q arm. This was question of need. Small arm is P arm and long arm is Q arm. P arm is also known as petit arm. Now, centromere is the point of attachment of two sister chromatids. Centromere breaks at anaphase to separate sister chromatids. Now, what is kinetochore? It is a trilaminar protein plate surrounding centromere. It has a high negative charge on it. Only negative charged piece placed at the centrophore, so chromatophore. It is the point of attachment for spindle fibers during cell division. Spindle fibers are nothing but microtubules which are attached, and see, this is the structure how the division of the central chromatophore occurs. Now here there are microtubules. These are attached by charge, positive negative to the poles. Now they at anaphase these spindle fibers do not condense, they shorten. Okay, shorten their length. So definitely if they would become shorter, then but the charge is not decreased, so definitely the pressure would be applied on these two ends and it would be get teared up. This part would go this side and this part would go this side. So that is somewhat how it is done. Now next, yes, it causes movement of two pairs on the chromosome like this. Okay, they get carried away. Now telomeres are those terminal ends at each chromatid made up of repetitive DNA, that is same type of DNA structure, something like this, again and again would be repeated. It is a type of junk DNA also. Now telomere is also called a non-sticky end, that is blunt end, because it has no unpaired nucleotides and therefore it is non-reactive. What is regarding no unpaired? Okay, here there won't be any of those. Here there won't be any of those pro, places that would be left without being reacted. So since there wouldn't be any protein or anything that would be left unreacted, so it won't be able to react. Now see, if you see, then these we know that A joins with T and C joins with G. Okay, these protein join with the same alternative of each other, and if suppose their T and A are not present, so definitely this A and T would above A and T would try to combine with something, so that it would be reactive. But here no such paired unpaired nucleotide is found. And some chromosome may have secondary construction also called a satellite. This is secondary construction. Now satellite is poor in AT, that is cyanoadenosine. Hmm. Presence of SAT chromosome for nucleus helps in the synthesis of ribosome, and then ribosome we know that protein. So indirectly SAT helps in the for also helps in the synthesis of protein also, because it would be important for the nucleolus, since it may contain some second construction may contain nucleolus organizing regions, and definitely nucleus helps in the synthesis of ribosome, and then ribosome is protein factory also. So classification of chromosome on the basis of function is two types: autosome and allosomes. Autosome for vegetative function, allosome for sex determination. Now on the second basis, that is type and shape, we have four types of them. Now type, that is for the type we use position of the centromere, okay, and for shape we use the anaphasic chromosome shape. Now Four types are metacentric, submetacentric. Now here in submetacentric, the centromere is in the center, exactly, and the shape of anaphase is V. In submetacentric, it is slightly away from the center. Here the shape is L. Now in acrocentric, the shape is very away from the center, so that one arm of each chromatin is very small. So here shape is G. Now in telocentric, there is actually no no lower arm. So here I perfect straight. Now study of chromosome. So staining of chromosomes, first of all. Now there are many methods. Banding method first. It is the differential staining that is not completely stained, means some parts of it is stained. Now in this method was developed by Caperson. He initially used the Fulgen stain. The four types of bonding methods: C banding, Q banding, and G banding and R banding. C banding, here C bands, that is constitutive heterochromatids, are stained. Most common method because it is used with specific species specific. Q banding is done by fluorescent rich um, cures, quinacrindine, and it stains A T rich regions. It is used for Y chromosome staining. G banding is also known as Gizma banding. It is used to color sulfur-rich protein of chromosome, and just opposite it R banding. Here it is also known as anti-Gizma because it is used to color sulfur deficient protein of chromosome. Now prokaryotype, karyotype, sorry, it is nothing but photograph of metaphoric chromosome. Adiagram is arrangement of metaphoric chromosome in decreasing order with respect to length of chromatophore thickness and length of chromosome and thickness of arm of it and position of the centromere. Example: human karyotype was developed by Denver system. Denver is a convention that is placed and it was done in 1961. According to it, Group A contain that is having 1 to 3 chromosomes is metacentric. Group B, that is has 4 and 5 is submetacentric. Group C, and that resembles to X chromosome, has from 6 to 12, that is submetacentric again. Now Group D has, that is 13, 14, 15, as acrocentric sad chromosomes. Group E has 16, which is metacentric, and 17, 18, that is submetacentric. Group F has 19, 20, that is acrocentric. Group G, that is Y chromosome, has 20 and 21, 22 and 22, that is metacentric side chromosome. Now, significance of karyotype is that it can be used for the identification and classification and evolutionary study. Telocentric chromosomes are primitive, and humans and apes' DNA match 99.9%. Their chromosomes have high degree of similarity, showing that apes are ancestors of man. Identifying gene disorders. Example like Down syndrome. Here, 21st chromosome present in it has 3